Ladies and gentlemen, welcome to Bioscience the musical. In the event of a fire alarm, please follow a student ambassador. Please turn off your mobile phones as the show is about to begin.
Hello, welcome all. Welcome to the inaugural York Scott Science Showcase. Today, we will witness the exciting, enthralling, enchanting researchers of York as they battle to win funding to support their research here at the University of York. The Longboy Memorial Grant provides support for lucky York researchers to work on whatever takes their fancy, thanks to the selfless donation from Longboy, our eminent university mascot. Our esteemed judge, Longboy Jr., will examine our scientists' pictures in detail and determine who will win this honourable and distinguished prize. I will also warn that you, the audience, will be voting for your personal favourite at the end. But first, here Longboy Jr. is to introduce himself. Thank you, Longboy Jr., for that captivating speech. And now, on with the show. Today, we will be hearing from a range of scientists from the biology department, working on topics as diverse as global diversity itself, through to the ways tiny molecules can influence diseases. First up, we're hearing from Rebecca Ruiz-Pavia about her master's project in the Skin Research Centre. Rebecca is researching the immunology of the skin, the tiny molecules that lead to skin diseases, and how skin cells respond to infections by bacteria. Her performance, Like a Prayer, some of you may recognise the name. Give it up for Rebecca!
Cells are a mystery, just see how bright they glow, adding bacteria, see how bright they glow.
When the specks appear, it's like a little prayer. I'm down on my knees, I want my master's please. When I hold the truth, it's like the holy truth, just like a prayer. You'll know I'll take you there.
Back in the lab, experimental science, biology, I need the key. Skin cells tell me, inflammasome, bacillus activated, protein in wounds, found in gut and skin, bacteria's toxic.
When the specks appear, it's like a little prayer. I'm down on my knees, I want my master's please. When I hold the truth, it's like the holy truth, just like a prayer. I will take you there.
All of my days are all quite slow but stressful. Care for these cells and organelles, they're all dying. I'll read a book, or maybe 2 or 3. I finally see the answer was me. I'm the key.
When the specks appear, it's like a little prayer. I'm down on my knees, I want my master's please. When I hold the truth, it's like the holy truth, just like a prayer. You know I'll take you there. When the specks appear, it's like a little prayer. I'm down on my knees, I want my master's please. When I hold the truth, it's like the holy truth, just like a prayer. You know I'll take you there.
especially if you can use those glowing proteins to understand them better. We're all especially excited about what the rest of this show has to offer after that unbelievable performance. Thank you, Rebecca.
Thank you.
Now, on to our next scientist. We'll be hearing from Davy Neefsey, who is a postdoctoral researcher in the lab of Dave Boucher, where he investigates immune cell responses to bacterial proteins and the tiny molecules called cytokines involved in these interactions. He's going to talk us through his work in his song, Firework.
Like immunity is attacking you instead of helping you. Do you ever feel inflammation right on your soft skin and it's uncomfortable? While I'm here to see if there's a solution. My mornings in the lab and dying tissue culturing. I infect the healthy cells with pathogens and they battle out. They make cytokines to light the blind, and variabilize lies into life. Pyroptosis is a firework, inflammation gone berserk. Make the pathogens flee, 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 as you regulate immunity. Pyroptosis is a firework, inflammation gone berserk. Make the pathogens flee, 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 as you regulate immunity. You don't have to feel all inflamed and sore. Some pharmacology can help those memories more, but inhibition can have adverse effects. And sometimes drugs cause harm as much as they protect. We need to take a beat, we need to take a breath, and think about the risks of all of that is cheap and death. Sometimes your cells they need to blow. When it's time you'll know. They make cytokines to light the blind. Their variabilize lies into life. Pyroptosis is a firework, inflammation gone berserk. Make the pathogens flee, 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 as you regulate immunity. Pyroptosis is a firework, inflammation gone berserk. Make the pathogens flee, 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 as we regulate immunity. Ow, 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 give me some drugs now, now, now. It's happening inside of you, you, and my research claims it's true. Pyroptosis is a firework, inflammation gone berserk. Make the pathogens flee, 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 as we regulate immunity. Pyroptosis is a firework, inflammation gone berserk. Make the pathogens flee, 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 as we regulate immunity. Ow, 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 give me some drugs now, now, now. Ow, 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 give me some drugs now, now, now.
Davey, that was a brilliant overview of your research on immune cell responses to infection. Pyroptosis certainly sounds like a rather wild process. Next up, we will be hearing from Violet Pepper about her PhD research in the Centre for Novel Agricultural Products. Her work is investigating the ways that mint plants can repel insects in her rendition of 360.
Violet, we're so glad you're here.
I was just about to start my song, actually.
Oh shoot, well, that's actually why we're here.
Yeah, we're looking for the next woman in STEM. Your song sounds really pretty, though. Can we focus on the content of my research?
You should be a little less assertive.
Actually, no. You should be a little more assertive. STEManism and all that.
Oh yes, we need to find our next STEM girlie!
Otherwise they're like, cease to exist, which is like, terrible.
Then, after that, we can totally hear your song.
Okay then, what about her? Babe, that's literally Kira Luffrey. Fine. What about her?
Totally scientist vibes!
I'm gonna start my song now. When I'm hungry and I made it, I'm your favorite breakfast lady. Call me Violet, I can and smile. Ooh, wet lips, dry lips, yeah, your name a myth makes me communicators. It's okay to still debate if this project is the one or not. Plant defense gets insects gone. Babysit media 12 years at once. We call it plant chemical weapon. We have fun. Jokes. Yeah, study mint leaves. Plant bugs biting, what do you see? Pesticides but eco-friendly. Used everywhere in agriculture. Like this mint leaves in a new leaf. Eucalyptus, lemon, yet the samples reap. They're everywhere, busy lab goes. Study that. Study that. I'll study that. That fresh plant is divine, shows compounds which are volatile. I grow the crops, code gene design, and it's stuck in your mind. My journey is so underrated. You can make it like I made it. If you love it, if you hate it, do what's me, has been David. Plant defense gets insects gone. Babysit, media 12 years at once. We call it plant chemical weapon. We have fun. Jokes. Yeah, study mint leaves. Compost and what do you see? Pesticides are eco-friendly. Use everywhere in agriculture. Like this mint leaves in a new leaf. You can just let me get the samples. They're everywhere, busy lab goes. Doing that, just reading that, just found out how so much, proving that, Violet, it's a wrap. Whoa,
Brilliant, thanks Violet. It turns out mint isn't as attractive to everyone as it is to me. We want to make sure that this award is as inclusive as possible and everyone has a shot at pitching their research for Long Boys Award. Does anyone in the audience have any research that they want to pitch? Oh! Yeah, you!
Yeah, can I?
Yeah, hi! Do you want to introduce yourself?
Yes.
Hello. Hi. I'm Ines. I'm a neurobiologist in the department. Yeah? Should we do it? Here I go again, pipette in my hand, fly embryos aligned. I follow the master plan.
I place them in the tube, filter and prepare, put medium in the mix, handle them with care. And then those neurons grow, if everything went right, connections fully form, reaching far and wide, paraformaldehyde locks each branching thread, keeps them all in place, just nerve cells in a dish. Looking through the microscope, how neurons form we want to know, understanding how a change in genes cause broken brains, turning left, signaling right, synapses fading over time, the signaling breaks down, top tangles form somehow. Will I find the cause below? Looking through the microscope. Under filtered light, I'm scanning all the frames, each axon traced in time. Not two are quite the same. And if we ever find the reason they decay, Alzheimer's might be cured, and flies might lead the way, with genes that match our own. Small brains, no cells clear, the building blocks are known. It is Drosophila. Looking through the microscope, how neurons form we want to know, understanding how a change in genes cause broken brains, turning left, signaling right, synapses fading over time, so signaling breaks down, to tangles for somehow, every color of the rainbow, secrets unlock when you look close. Will I find the cause below? Looking through the microscope.
Wow, what incredible research you're doing, Ines. Fascinating that we can see how individual cells work with those fancy microscopes. Amazing how you can use a fly to understand the inner workings of the brain. Next up. Next up, we have an ecologist who has recently finished her PhD and is now an associate lecturer at the University of York, teaching all our wonderful undergraduate students about conservation. Lou Mamalis will be performing her song, Buffer Buffalo, to tell us about her research into the endangered ibis bird in Cambodia and how we can conserve this important animal.
Help the ibis last the day,
Day is sad.
and still there never seems to be an ibis here for me to see.
That's too bad.
In my dreams, I have a plan to save the ibis from extinction as we kill their homes and take the trees. The water's dry, there's no plan B.
Buffer, buffalo, we need you so to save this cool old world. Buffer, buffalo,
I think I know this is a cool old world. All the things I could do old world. It's a cruel old world. I traveled far to southern Asia. What I found will quite amaze ya.
What did you find?
Buffalo, they stand and wallow, maintain wetlands, mud and water.
They're one of a kind.
So I must leave, I'll have to go to Cambodia for the buffalo. To do this first, I must confess, I've got a plan, I've done the tests. Buffalo, we need you so to save the wider world. Buffalo, help nature grow, create a better world. The things I could do if we had more buffalo, create a better world. Buffalo, we need you so to save the heron's world. Buffalo, give storks a home, help populations grow. The things I could do with a herd of buffalo, create a better world. We could save the world.
Wow, thank you so much, Lou, for that engaging look into the world of the ibis. It's so important to keep these animals in their habitats, and so fascinating how interacting with buffalo can help them.
Thank you.
Next up, we have Jack Hatfield, a computational biologist in the Leverhulme Centre for Anthropocene Biodiversity. He's going to tell us about his work researching global biodiversity change in his song, I'm Just Jack.
Doesn't seem to matter what I do. Some species won't make it through. There are patterns to be found, but I know they're really hard to explain, driving me insane. All life is changing really fast, quicker than the past. 'Cause I'm just Jack, trying to bring biodiversity back. It is my destiny to plant and find insects for your high curiosity. I'm just Jack, you see data, I see a trend. What will it take for them to see the loss of vision lost through history? About this world, the change it's making. Is it our fault we're not improved or understanding? These places have become less diverse for exploring. I'm a scientist. Biodiversity
just Jack, and the species I keep track. It is the complexity of my ecosystems that fascinates me. I'm just Jack, where I see life there's always death. Or will it take for them to see the loss of Bishop Moss through history? I'm just Jack, is that enough? And I'm good at ecology stuff. So hey, check out my work, I'm just Jack. My name's Jack, and so am I. Some species are in decline. So hey world, check me out, I'm just Jack. Baby, I'm just Jack.
That was incredible, thanks Jack. An important topic to analyse as global factors such as climate change disrupt our delicate ecosystems. That's carrot. And now we have Lily Cabrera Cosme and Charlotte Titterington Coleman, two students at the Centre for Blood Research, who will be singing about stem cell biology, clonal evolution, and the role of inflammation on stem cell function. And alongside all the time and effort she and Lily dedicate to their research project, Charlotte will also be working as a princess at Disneyland Paris this summer. Therefore, paying tribute to life outside the lab, comes a Moana medley titled, What Do They Know?
I've been staring at these stem cells all day, all night, as long as I remember, and I'll tell you all the reasons why. I wish I could get these daughter cells to treat blood cancer. Do you know how hard I try? Every step I make, every case I track, every look we take will not lead us back to the place we know. Why not try and go elsewhere and see? It's less invasive in the chest, the sternum, it calls me. We can explore how far this goes. Produce cells for our health, stop disease. If we find these, what will this show? If we know, there's just no telling how far we'll go. Charlotte deserves recognition, gratitude for her work. She's the best in looks. Get what Lily does for us. She's worked up to her neck. Sign her check. When they speak of it, they research their word, they go on and on. Anything diagnostic phenomenon. Transplants, we have extra. They call them monsters, cause look what they found. Return, begin, we'll tell you what for. Princess at Disney, yeah, pop. What's the lesson? What is the takeaway? Child, there's a lot that he rocks, you better make way. Beneath our bones, and they have yourselves. Yeah! People are great and they say you're welcome. You're welcome. You're giving the world some hope. Hey, it's okay, you're welcome. You're welcome. Back to the lab again, I gotta go. Working all day and night. You're welcome. You're welcome. Cause I'm gonna need those funds. But how they behave, life saved. You're welcome. You're welcome. Stem cells can do anything that it goes. You're welcome. You're welcome. You're welcome. And thank you.
Thank you, Lily and Charlotte. What exciting work you're both doing. Truly incredible to learn more about about how stem cells differ in different tissues. Their vital role in various diseases is so important, but also really fascinating to hear about. Thank you. Now, we will hear from Victoria Hill, a postdoctoral researcher in the Alfie Lab, who has made some time in her busy calendar to tell us about her incredible work on mosquito genetic engineering. Her song, Busy Postdoc, will tell us all about it.
Mature, experienced and rational, except when I get hit with a deadline. We're saving lives, we're just really ethical, trying to stop the killer mosquito. I'll go beyond and above. My lacquerton covered over to his place, and I'm wearing my blue gloves. I didn't want an early night now anyway. Yeah, I'm a busy woman. I wouldn't have any space in my calendar anytime. But if you want my methods, I'll be your perfect postdoc till the day turns into the night.
Busy postdoc all the time. Cut the mile and the mosquitoes can fly.
Lots of mosquitoes, we need them to not fly, spreading diseases like Zika, fevers, dengue. Just stop the girls, yeah, they are the ones that bite. CRISPR-Cas9 passed down through the males. If they don't reproduce, I'll just deem them gay. Now I don't need my gloves. We get the blood from horses, there's really no waste. And after they feed, they lay eggs. We hatch the eggs, grow them up, pick the bouquet. Yeah, I'm a busy woman. I wouldn't have any space in my calendar anytime. But if you want my methods, I'll be your perfect postdoc till the day turns into the night.
Busy postdoc all the time. Cut the mile and the mosquitoes can fly. When they can fly, they can bite. Expressing Cas9.
Get the timing right. Now I don't need my gloves. My lacquerton, I'm finally leaving this place. But wait, I still need my gloves. Forgot to pick out this last little bouquet. Yeah, I'm a busy woman. I wouldn't have any space in my calendar anytime. But if you want my research, I'll be your perfect postdoc till the day the mosquitoes all die.
Holy moly, that's a complex bit of work. Well done, Victoria, for explaining all that to us so well. Mosquitoes sure are a pest, and we are glad you're tackling them in your research.
Thank you.
She's sure busy though. Bye, Victoria. Not sure how you found time to come and tell us that today. Our final early career researcher is Federico Sabadin, a lecturer in the Centre for Novel Agricultural Products, to tell us all about his work to discover new proteins that we can use for a huge range of different applications in his song, Plant Waste.
I am not throwing away plant waste. I am not throwing away plant waste. And you are focused on the mission. I'm stressed, I'm structured, and I'm not throwing away plant waste. I'm wanna turn the carbs into simple sugars. I probably shouldn't write, but hey, I practice and polish. The problem is they burn plant-based waste, and that's foolish. I gotta research just to make my break. With every mistake I drop knowledge, I'ma tell you how it works. A simple bit of facts. Starting at the top, the carbs are locked in tightly packed packs, found in cell walls at the edge of the cell. But you see, they're hard to expel. I rebel. Every burden, every disadvantage I have learned to manage with companies that let smog and trash do damage. The plan is to find an enzyme we can use to break down the carb. But I must spell out my name. I'm the FEDERICO. So we are meant to be a carbon neutral economy. Meanwhile, Brazil's cars use bioenergy. Essentially, that fuels green technology. Using sugarcane to avoid a spending spree. We ain't ever gonna be emission free. But I'll do my research in this century, and to me,
He says in parentheses,
enzymes make biomass, bioenergy. Why would people invest in what sets them free? One day you'll hear of my journey, and I'm not throwing away plant waste. I am not throwing away plant waste. Ayo, I'm focused on the mission. I'm strict, stubborn, and structured, and I'm not throwing away plant waste. I am not throwing away plant waste. I am not throwing away plant waste. Ayo, I'm focused on the mission. I'm strict, stubborn, and structured, and I'm not throwing away plant waste. It's time to break it down. I try my best working on polymers, observing insects, sugar demolishers, see how they digest their food to give me a clue what to do. When I get a setback, I'ma break it down. I came to York from Italy. Can't you see why this all means a lot to me? I'm researching science cause I know it's my chance to advance society. Instead of letting it burn, I'ma break it down. Now that the simple sugar's free, it's time we get the government to agree to see. It's you and I, do or die. Wait till I shoot my shot like a catapult with my newfound result. I'ma break it down. Penises, lower your voices. I'ma take this time to bust out a rhyme and explain to you why we can't let this fly. It's do or die. You gotta keep it simple. It's simple. You gotta break it down. Now see what we've got. Scientists working, looking at data non-stop. I think this crop's got gold. Sugar's locked in the stock. Let's hatch a plot louder than the tick of the climate's clock. What are the odds? The gods would favor breaking the crop. Challenging norms by using enzymes as our chemical stock. We're turning waste into something fossil fuels never unlocked. Give me a position. Show me where the information is. Oh, am I talking too loud? Sometimes I get overexcited, shoot off at the mouth. I never had a group of people listen before. I promise that I'll make you all proud.
Let's get this guy in front of a crowd!
I am not throwing away plant waste. I am not throwing away plant waste. Hey, I'm focused on the mission. I'm strict, stubborn, and structured, and I'm not throwing away plant waste. I am not throwing away plant waste. I am not throwing away plant waste. Hey, I'm focused on my mission. I'm strict, stubborn, and structured, and I'm not throwing away plant. Not throwing away plant. Not throwing away plant. Not throwing away plant waste.
Thank you, Federico, for that wonderful performance. I'm sure our audience are as excited about enzymes and their potential to break down complex sugars as you now. Our esteemed judge will now review the work we've heard about and give his verdict on the winner of the Grand Longboy Memorial Prize. However, while he deliberates, we have a special guest. Professor Paul Kay, world-leading expert in Leishmaniasis immunology and a long-standing member of the University of York community since he joined in 2004, is here to tell us all about some of the fascinating work he and others are doing at York on the Leishmania parasite and the disease it causes, Leishmaniasis, in his song, Talkin' About Leishmania.
Parasites inside our cells.
Talking about Leishmania.
Patient scoring, kids unwell.
Talking about Leishmania.
Disease buns, poor nation.
Talking about Leishmania.
Immune system dysregulation.
Talking about
This is our fascination. It's time we're facing, yeah. Making vaccines is hard today.
Talking about Leishmania.
How do we keep that leash away.
Talking about Leishmania.
Sandflies spreading infection.
Talking about Leishmania.
Differences in disease progression.
Talking about
This is my fascination. This is my motivation, yeah. Some in liver, some in skin.
Talking about Leishmania.
Holistic view is how we win.
Talking about Leishmania.
Human challenges and vaccine trials.
Talking about Leishmania.
International collabs spanning miles.
Talking about
This is my
Leishmania.
fascination. This is my motivation, yeah. My fascination. My motivation. My fascination. 79 is when us started.
Talking about Leishmania.
In the lab is where my heart is.
Talking about Leishmania.
Nowadays I run the team.
Talking about Leishmania.
Curing parasites is my dream.
Talking about
This is my
Leishmania.
fascination. This is my motivation, yeah.
Incredible! Thank you, Paul, for that journey through the world of Leishmania parasites and how they cause disease. It must be tremendously tricky to research something like that, and to have developed a human challenge model is truly remarkable. Vaccine trials, treatment approaches, and an in-depth understanding of how the parasite interacts with the people it infects and sandflies that spread it. Breathtaking research happening right here at the university. Audience, I think we should give it up for the parasitology community here at York. And now, the moment everyone's been waiting for, your chance to vote. We're going to do it like this. You can vote as many times as you would like, but we're going to ask that you vote by cheering. So, let Longboy Jr. know whose research you think he should fund. Like a prayer. Firework. 360. Microscope. Buffer Buffalo. I'm just Jack.
Oh my God!
What do they know? Busy postdoc. And plant waste. Well, it sounds like you really enjoyed them all, and I really couldn't tell which was everyone's favourite. But just on time, I've had news that Longboy Jr. has reached his verdict. We will go to him now. Well, you heard it here first, folks. Longboy Jr. has decided that everyone's research is too valuable and important to miss out on, and he simply couldn't fund just one project. We'll be funding everyone's research! Congratulations to all of our researchers. We can't wait to see what you do in the future and the impact your research will have in your different fields. Thank you so much for telling us all about it today. Audience, give it up for our incredible researchers, both those here today and everyone else here at the University of York doing vital research that's happening.